everyone, welcome to Biomix Lab. Today in this video, we are going to discuss another one topic that is a differential centrifugation. We can call also it as cell fractionation, centrifugation. It is known as cell fractionation centrifugation because in the cell we are having various type of cell organelles present. By using such type of technique, we can differentiate the cell organelles or we can separate the cell organelles. So what is the general procedure of this differential centrifugation? So in general, what we have is we are having a sample. What happens in case of a sample? What happens in a differential centrifugation? If we are having some of the sample contains solutions, and in the solution there are large particles as well as small particles present. So when we want to separate these particles, large particles and small particles, what we have to do is we have to do a centrifugation. So according to the weight, they can be separated out. Like those who are heavy or those who are large in the solution, they will be settled down and they are called as pellet. And those who are small or which are light, they will be in the solution or they will be present in the upper part, that is known as supernatant. Then we can separate it out, like we can take this supernatant in one tube and the pellet in one tube. Whatever we want to do our experiment, we can use this. So for the centrifugation, for this separation, mostly the sucrose solution is used. How can we see that? How can we use a sucrose solution for measuring our concentration? Like the 10%, 20%, 30% sucrose solution are being used for doing the centrifugation, and they are settled down according to their concentration or according to their weight. The suppose we are having a 50% solution of sucrose, that means it is dense. If it is dense, it will be settled down, and if it is having 10% of the sucrose, that means it is least dense, it is having low weight, so it will be settled up. In this way we can say here that it is 1.5 gram per centimeter. Okay, so it is having high molecular weight or it is dense, that is the reason it is settled down. And here we can see that it is having 1.09, that means it is less dense, that is the reason it is settled in the upper portion. So whenever we are using some of the substances or some of the solutions for separating, they will be this sample will be placed in the upper part. When we are doing the centrifugation, according to the gravity or according to their dense, they will be separated out. So here we can see that the lysosome which is present, it is having 1.12 gram per centimeter cube. Its weight is present. So here they will be present. The next is the mitochondria. This mitochondria is having 1.18 gram per cm cube. That means its place is here. It will be present here, and this is the sucrose solution. This is the sucrose solution. Okay, and according to the gravity, it is being arranged. This is our control. Like we know that in which area how much concentration is present. Accordingly, we can extract our particles which is needed or the organelles which is needed. So how they, let us discuss now why it is also known as cell fractionation technique. So we are having a cell, and in the cell there are various type of cell organelles present. So what we have to do is we have to separate the cell organelles. For this, what we have to do first, we have to disrupt this cell. So when we are disrupting the cell, there are plasma membranes as well as plasma membrane of ER is present. So this will not be in intact form because we are giving, for this disruption, we are giving some of ultrasonic waves, like we can also give some of the osmotic pressure, or we can simply blend this for breaking. So whenever we are doing this, the plasma membrane as well as the ER membrane, they will not be in the intact form, they will be in small fragments, and the small fragments can convert into small vesicle like structures. There are also some intact organelles, like it can be a Golgi, it can be ribosome, it can be peroxisome, it can be mitochondria. So these are some of the intact organelles which are present in the cell, and we want to isolate the, suppose we want to isolate the Golgi. So this cell organelles which are present, they are of different molecular weight. That means those who are having high molecular weight, they will be settled down, and those who are having low molecular weight, they will be present in the supernatant. So let us see how this is done. So we have to take like as we have already discussed that we can use a sucrose solution for the separation. Here also such type of solutions can be taken, and filter homogeneate to remove clumps of unbroken cells and connective tissues. That means if we are having some unnecessary things, suppose we want to separate some of the organelles and there are some of the unnecessary things present in the solution. So first thing what we have to do, we have to separate them out. So this is the homogeneate, filtered homogeneate, which is present. That means our sample is present in this solution. Then what we have to do for the separating the organelles, we have to centrifuge it. When we are centrifuging, first what we want to do is we have to centrifuge in low gravity or low RPM. We have to centrifuge for less time. So in the first case, let us say that it is centrifuged in 600 G for 10 minutes. So the nuclei is separated out because this nuclei is heavier than any other organelles. Okay. Now, as it is separated out, it is present in the lower end. That means it is working as a pellet. It is as it is having the high molecular weight. And the other things of the cell, that is the organelles, it may be a vesicle, they are present in this supernatant. This part is known as the supernatant. Suppose if we want to work with the nuclei, we can separate the supernatant and we can collect the nuclei and perform our work, and we can stop our experiment here for the isolation of other organelles. But what if we want to do some of the experiment in Golgi? We want the Golgi to be isolated. We have to collect this supernatant in another tube, and we have to centrifuge them with high RPM, that is 15,000 G for 5 minutes. Here the rotation is more. So now what we can get? We can get the mitochondria, chloroplast, lysosome and peroxisome in the pellet. These are present in the pellet in different layers. So now if we want some other organelles to do our work, we don't require mitochondria, chloroplast, lysosome and peroxisome. For our work, we need something else, like it may be a plasma membrane or some of the fragments, or it may be a cytoplasm. We require only the cytoplasm. So in that case, we have to take this into another one tube and we have to do the centrifugation. So the next centrifugation is done for 1 lakh for 60 minutes. If we do it, we are getting the plasma membrane, microsomal fraction, fragments of ER and last polysome. Because these fragments which are present, they are having low molecular weight. That is the reason we have to give them more time, and in more RPM when we centrifuge it, we can get this. So next, if we want some of the ribosomal subunits for our work, so in that case we are not getting, till now we have not got any of the ribosomes. So we have to take the solution from here to another tube, and we have to centrifuge it for 2 hours in 3 lakh G. Next, if we want only the cytoplasm, then we have to pour this solution and we have to centrifuge out. Okay, so in this way we can get different type of cell organelles. So at first what we got in 600 G for 10 minutes, we got the nuclei. At 15,000 G for 5 minutes, we got mitochondria, chloroplast, lysosome and peroxisome. Next, for 1 lakh 60 minutes, we got plasma membrane, microsomal fraction, fragments of ER, large polysome. And in 3 lakh for 2 hours, what we got? We got ribosomal subunit in the pellet, and in the upper part, that is a supernatant, the cytoplasm will be present. In this way, the cell fractionation technique is used for separation of the cell organelles. This was all about today's video. Thank you for watching this video till the end. And if you like this video, please do like, share, and subscribe. Thank you.